Okay, good evening everyone. Good evening. Good evening everyone. Good evening friends. I hope everyone is doing good. Great. Okay. So actually morning times I'm having this great power problem even though we are working with inverter due to continuous power work I mean say doing due to continuous power cut. I'm unable to take the classes on the morning. So, but don't worry at all. We will continue in the morning time only from tomorrow onwards. But if anything to be rescheduled, I will inform prior itself. By around 4:30 or 4:45 a.m. only, I will inform every one of you. Okay, so friends, let's continue the hormone. See, I hope every student remember friends in the last class. Good evening everyone. In the last class we have discussed about the hormone auxin. We have completed the hormone auxin, as well as friends we have completed the hormone gibberellic acid also. Good evening all. So friends, now what we are left with? We are left with the hormone cytokinin, we are left with the hormone ethylene. At the same time friends, we are left with the hormone abscisic acid, right? So let's try to finish the hormones. How many hormones we can finish in this particular class itself? Okay, let's talk about the cytokinins. So friends, please kindly don't interrupt me for the period of 15 to 20 minutes. I will explain the hormone cytokinin very elaboratively. Just pay attention continuously and keep on listening me for 20 minutes. Okay, let's start. So friends, when we are understanding the cytokinin, I want every student has to remember one common point. What is that common point? If this, listen to me carefully, if the hormone cytokinin wants to work effectively in the plant body, compulsory there should be a presence of auxin. Once again, listen to me every one of you. The hormone cytokinin work very effectively in the presence of auxin. Now friends, let me tell you all of you how exactly the hormone cytokinin were discovered actually. You know what the scientist does? Just listen to me friends carefully. Scientist has taken three mixtures. What are that mixtures? First they have taken a coconut mixture, coconut milk mixture, coconut milk, or the second one they have taken DNA extract. Friends, listen to me carefully. And friends, they have taken vascular extract. Friends, here the word vascular extract means friends xylem extract. Vascular extract means what friends? Xylem extract they have taken. Every student listen to me very carefully. They have taken these three mixtures friends, and these three mixtures they have added to the auxin. Every one of you understand friends? They have taken these three mixtures and the three mixtures they have added to the auxin. Now this entire mixture, you know what they have done? They introduced into the internodal region of the stem of the tobacco plant. Every one of you listen to me friends. This is the tobacco plant. These are all the nodes. In between, what are there my friends? Internodes. These are all internodes. Friends, listen to me carefully once again every one of you. It's very simple. All of you focus. They have taken these three mixtures and they combined with the auxin, and this entire mixture they have introduced into the internodal region of the stem of the tobacco plant. Then what they observed? They observed the division. What they observed friends? They observed the division. Every student listen to me. Now what is the conclusion friends? Everyone understand? Among these three mixtures, any of these mixture contain a chemical. Once again understand, among these three mixtures, any of the mixture contain a chemical. When that particular chemical combined with the hormone auxin, it is showing basically a division. So friends, what is that particular chemical? Every student try to understand friends whatever I am writing here. Miller is the scientist in the year 1955. He has isolated the first cytokinin. You know friends, from where he has isolated the cytokinin? He has isolated the cytokinin from the herring sperm DNA. Okay, herring sperm DNA. Do not forget the point at all. And that particular cytokinin, herring sperm is the name of the fish friends. Don't get confused. What is herring sperm? Herring sperm is the name of fish. From the fish, he has isolated the first cytokinin. And you know what is the name he has given for the cytokinin? He has given the name kinetin. But friends, every student remember, this is not the natural cytokinin. Please listen for the exam point of view, especially for part B. The same scientist Miller only, he is the one who isolated the natural cytokinin. From where he has isolated this particular natural cytokinin? From the endosperm of the corn. From the endosperm of the corn, he has isolated as natural cytokinin and he has given the name giatin. Friends, please kindly don't confuse. The naturally occurring cytokinin is called as giaxanthin. Anyway, while reading the abscisic acid, I will tell you anyway, but listen, please kindly don't confuse. Naturally occurring abscisic acid, we are going to read it. Naturally occurring abscisic acid is the giaxanthin, and naturally occurring the cytokinin is what, my friends? Zeatin. Please kindly don't confuse between these two. And you people, no need to remember the structures at all. I have already told you. What I want you people to remember here is that cytokinins are, listen to me carefully. Cytokinins are basically derived from what, friends? Purines. Especially from where? Especially the cytokinin, friends, is derived from the adenine. That's why we can call it aminopurine. So friends, what is the best method I have suggested for all of you while remembering the hormones? While remembering the hormones, you have to compare one hormone with the another hormone. When you compare one hormone with another hormone, trust me, you can remember it very easily. Now let's try to understand. Keep on comparing whatever we studied in that particular hormone. If you compare here, it will be very easy. Let's quickly recall. When we are studying about the auxin, what we studied, my friends, we read very clearly hormone auxin is the rooting hormone. Tell me everyone, why do we call the auxin as the rooting hormone? Does it is synthesizing in the root? Not at all. Auxin synthesized in the shoot. From the shoot, auxin travels to the root and it is going to show their major mechanism in the root. That's why we are calling auxin as the rooting hormone. But my friends, whenever you heard the name of a cytokinin, one of the common point has to remember, cytokinin is the shooting hormone. What do you mean by that, friends? Cytokinin is synthesized in the shoot. From the shoot, cytokinin is traveling to the, I'm so sorry friends, cytokinin synthesized in the root. From the root, it will travel to the shoot and it show their mechanism of action. Listen, cytokinin synthesized in the root travel to the shoot and it is involved in the development of a shoot growth. Please kindly don't confuse. And most important point, my friends, how the cytokinin start moving from the root to shoot, friends, that is an important statement. Cytokinin, whatever it is synthesized in the root via xylem. You know, right? Xylem is the one which is involved in the movement of a water, ions, all these. So that particular cytokinin from the root, friends, it starts traveling to the shoot, and once it reaches the shoot, it will be helpful in what, friends? It will be helpful exclusively in the division. It will be helpful exclusively in the division. Please kindly don't forget that. And everyone listen my statements carefully, friends. You people no need to remember the structure at all. Please kindly do not worry about that. You people no need to remember any kind of a structure. But I want you people to understand the pathway. You don't need to remember the structure, but understand the schematic presentation of the pathway. Don't forget that. And every student, once again, I'm reminding for all of you friends, I hope all of you remember. When we read the auxin, I have told you very clearly to all of you, tryptophan is the major precursor for the auxin. And we all remembered when we read the gibberellic acid, what we read, my friends, please listen to me. We read GGPP is the major precursor for the gibberellic acid. Friends, whenever you are reading the cytokinin, all the students listen to me carefully. DMAPP. What was that? DMAPP, dimethyl allyl diphosphate or pyrophosphate. Dimethyl allyl pyrophosphate is the precursor for the synthesis of this particular cytokinin. How many carbon containing compound cytokinin is, friends? Cytokinin is just 5 carbon containing compound only. Please kindly don't forget the statement. I will be explaining you elaboratively. Just look into the diagram very carefully. Just listen my words. And what is the function of this particular DMAPP? The ultimate function of the DMAPP is what, friends? Donor of the isopentenyl group. Every one of you listen to me very carefully, friends. DMAPP can act as a donor of the isopentenyl group. Everyone listen to me. It is donating the isopentenyl group to whom? All of you listen. It is donating the isopentenyl group to the AMP. Listen to me carefully. Upon receiving the isopentenyl group, AMP converted into IPMP. What is IPMP? Isopentenyl monophosphate. Very simple, my friends. All the students look into this. This is the AMP, friends. Now AMP is added by what group? Isopentenyl group. Upon adding the isopentenyl group, we are just calling it as the isopentenyl monophosphate. That's all. Now every student listen to me carefully. From this particular isopentenyl monophosphate, what we have done, you look into this. I have removed this phosphate. Upon removing the phosphate, isopentenyl monophosphate, friends, it is going to converted into IPA. What is the full form of IPA? Isopentenyl adenosine. Now friends, this particular isopentenyl adenosine, upon removal of the sugar, that is ribose, it is ultimately converted into what, friends? IP, isopentenyl adenine. Now this particular isopentenyl adenine can be treated by a specific hydroxylase enzyme. It is giving rise to the transghiatin. Friends, it's very simple. Now transghiatin can be treated by an enzyme called as a cis-trans isomerase enzyme, and obviously it is going to convert it into cisghiatin. I am explaining for all of you once again. Please look into this very carefully. Who is the donor? First, who is the precursor? AMP. What is the major starting compound? DMAPP. What did I tell to all of you? DMAPP is basically donating the isopentenyl group. Upon donating the isopentenyl group to the AMP, AMP will become IPMP. Now IPMP, upon removing the phosphate group, we converted into IPA, which means isopentenyl adenosine. From this particular isopentenyl adenosine, upon removing the sugar, it is converted into IP. We can call it isopentenyl adenine. Now this isopentenyl adenine, upon treating with the unknown hydroxylase enzyme, it is converted into a transghiatin. The transghiatin can be treated by a cis-trans isomerase enzyme. Ultimately, friends, it is converted into cisghiatin. And what about this? This is occur in the bacteria. You people no need to remember at all. You have to remember whichever is happening in the plant only. What is the major precursor? DMAPP. It's a very easy. You don't need to struggle for this. Okay, 99.99%. I am talking exclusively about the cytokinin. You can expect the questions in the cytokinin either from the functions or, friends, from the signaling. These are all the two areas where we can expect the question. So now we are discussing about the function. I am requesting every student to listen to me very carefully. We should not miss even a single function also. And signaling. Signaling is little more confused. So that's why I am requesting everyone pay utmost attention. Okay. What is the ultimate function of the cytokinin? It is regulating the cell division in the shoot also, in the root also. Every student has to listen my statements in a simple way. Cytokinin, where it is synthesized, friends? Cytokinin synthesized in the root, travel to the shoot. Then undoubtedly, my friends, what it is doing? It is showing the positive growth. Where? In the shoot. Undoubtedly. And where it is showing the negative growth? in the root. Why it will show the negative growth in the root? Because it is synthesized in the root only. Then how it can show the action? It cannot show the action, right? But every student has to remember, suppose example, there is, listen to me carefully, suppose example, there is a over expression of the cytokinin genes or mutation in the cytokinin oxidase gene. You know friends, what is happening? Opposite things are happening, which means positive growth occur in the root and negative growth occur in the shoot. This is happened only when there is a mutation. Every one of you look into this carefully, friends. Listen to me carefully. Cytokinin will show the positive growth in the shoot apical meristem, negative growth in the root apical meristem. But what is the point you people has to remember? Over expression of the cytokinin oxidase gene, which leads to the enhancement of the root growth. So you have to remember, cytokinin will increase the root growth only under a condition where the cytokinin expressive genes got mutated. Otherwise it is not going to show that particular action. Now that is how cytokinin is involved in the cell division. And friends, I want you people to remember one more very unique statement. Cytokinin also involved in the activation of a D type of cyclins. Everyone just listen to me carefully. If the D type of cyclin wants to be activated, first there should be a gene called as CYCD3 gene. This gene has to be expressed. If this CYCD3 gene wants to be expressed, we have to activate the CDC2 kinase. If the CDC2 kinase wants to be activated, we have to activate the CDC25 like phosphatase. Every one of you listen to me very, very carefully. What the cytokinin is doing, friends? Cytokinin activate CDC25. CDC25 in turn activates CDC2 kinase. CDC2 kinase in turn activates CYCD3 gene. CYCD3 gene automatically code for the D type of cyclin. This is involved in the cell cycle regulation. In a simple line, cytokinin is involved in the cell cycle regulation by activating a D type of cyclin. Very, very simple point it is. That is the first function. What is the first function? Cytokinin show the positive growth in the shoot apical meristem, negative growth in the root apical meristem. Now friends, here comes the most important, nearly twice asked questions in the part B in our CSIR exam. Friends, all of you listen my statements carefully without having a confusion. Please try to recall. I told you very clearly, ethylene is the hormone. We are going to read next. Ethylene is the hormone which will promote the senescence. Is it or not? Ethylene is the hormone which will promote the senescence. Friends, what do you mean by the word promoting the senescence? Which means it is making the leaf to become older. That is what we read. But friends, every student listen my words carefully. Cytokinin will do the opposite action. What the cytokinin is doing, friends? Cytokinin will cause delay in the leaf senescence, which means, listen to me, cytokinin never ever allow the younger leaf to become older. I mean to say, it never ever allow the leaf to turn into yellow. Cytokinin always help the younger leaf to become green color for the longer time. First of all, every student has to remember one unique point. You can ask me, sir, first of all tell me why the leaf turns yellow from the green. That is my question. How does leaf is going to converted from the green to the yellow? Every one of you listen. The reason is very simple, right? The reason is loss of chlorophyll. This is the first and most important reason. Loss of chlorophyll, and friends, loss of RNA. At the same time, friends, loss of protein. Every student, please listen to me carefully. What, friends? Loss of chlorophyll, loss of RNA, loss of protein. All these will cause the leaf to turn into yellow. Now you can ask me a question. Sir, how this particular chlorophyll, RNA, protein, they can be completely lost? Because of certain inhibitory enzymes. Very simple point. Everyone listen to me carefully. Certain inhibitory enzymes, what they will do? They degrade the chlorophyll, they degrade the RNA, they degrade the protein, which are required for the leaf to make it green for the longer time. When all these got inhibited, what is happening? Obviously, leaf turns into yellow very fast. But my friends, what is the point? You people has to remember, whenever the cytokinin is present in the plant body, every one of you listen, cytokinin will correct reins, which we call chlorosis, loss of chlorophyll. Cytokinin inhibit this degradative enzymes. Simple. Cytokinin will degrade or inactivate. Actually, cytokinin inactivate the inhibitory enzymes. When the inhibitory enzymes get inactivated, there is a no loss of chlorophyll, no loss of RNA, no loss of protein. Then the leaf never turns into yellow. Leaf will be remains green color for the longer time. What the scientist does, they have done a small experiment. You know, friends, what they have done? They have taken some green color leaves, and they have taken actually green color leaves. Half of the green color leaves they kept in the cytokinin solution. Half of the green color leaves they kept in the normal solution where the cytokinin is not present. Every student listen, whatever the leaves that are kept in the cytokinin solution, they remain green color for the long period of time. Simple point. They remain green for the long period of time. Whatever the leaves that kept in the normal solution where there is no cytokinin, what is happening, my friends? It is converted into yellow. So what is the point you have to remember? Listen, I am summarizing the statement. If the cytokinin is present in the plant body in an adequate amount, it always try to delay the leaf senescence. It never ever yellow the younger leaf, not to convert into a older very faster. That is the reason we can use a word, cytokinin is the hormone which delay the senescence. And friends, this experiment was done by a scientist called as Richmond Langerhans. That's why this effect also we can call it as a Richmond-Langerhans effect. Please kindly don't forget that. Here comes the unique function. I hope every one of you remember, whenever we are studying the hormone auxin, we clearly read that hormone auxin always promote the apical dominance. I hope every student remember. What do you mean by the word promoting the apical dominance? I told you, when auxin is present at the tip of the shoot, what it is doing, my friends, it never ever allow the growth of the lateral branches. That is what I have explained to you in the auxin, which means it only allow height to occur. It will only allow height. It only allow height to grow. It never increase the girth, or it never allow the branches to grow. But friends, every one of you listen, auxin is promoting the apical dominance. What the cytokinin is doing, friends? Cytokinin is doing just the opposite function. Cytokinin inhibit the apical dominance, which means, my friends, cytokinin will go on to dominate the level of auxin. As a result, what happens, friends? The lateral branches or the lateral buds can grow very easily. So friends, that is why, what is the statement I can give to all of you? I can give a statement that cytokinin and auxin both are antagonistic to each other. Both are antagonistic to each other only in terms of apical dominance. Because auxin is never yellowing the growth of lateral branches, cytokinin is always yellowing the growth of the lateral branches. That's why we can call it both are acting oppositely, antagonistic in nature. Next, most important function, this is also repeated twice in the exam. Direct question. Every one of you listen. Morphogenesis, which means the development of a root and shoot growth. Every students try to understand. Suppose I have taken a culture. In the culture, what I have done? In the culture, friends, I kept a high auxin ratio and I kept low cytokinin ratio. Friends, when I keep high auxin and low cytokinin, here who is present in the higher concentration? Auxin. I already told you very clearly. Auxin majorly show their mechanism of action where, my friends? Root. Since the auxin concentration is more, obviously friends, it leads to the development of a root growth. Now what I have done in the culture, I kept low auxin and friends high cytokinin. So here, correct, you can, very good Devesh, you can call it exactly anti-auxin hormone, correct, exactly. So low auxin and I kept a high cytokinin. Now what you have to remember, wherever the cytokinin is abundantly acting, obviously it leads to the development of a shoot. And most of the students will think that in the culture, when you keep auxin and cytokinin in the 1:1 ratio, most of the students will think that root also will develop, shoot also will develop. That is absolutely wrong statement. When auxin and cytokinin will be present, I mean to say, when both are present, every student listen, it leads to the group of cells. It leads to the mass of cells. It leads to the bulk of undifferentiated cell. What, my friends? Undifferentiated cell. What do you call that particular undifferentiated cell, friends? Because they cannot give rise to root. They cannot give rise to shoot. That's the reason I can call it undifferentiated cell. When this callus is forming, friends, the callus is forming only when the auxin and cytokinin are present, only in the 1:1 ratio. That's the unique statement. Suppose example, I kept auxin as the 2 ratio and cytokinin is the 1 ratio. Then what happened? Obviously it will leads to the development of a root. Suppose I kept the auxin in the 2 ratio and cytokinin I kept in the 4 ratio. Obviously what happened? It leads to the development of a shoot. But if I keep both auxin and cytokinin both in 1:1 ratio, it never leads to the development of any root or shoot. Instead, it is going to give rise to the formation of a callus. That's a so simple thing you can remember. That is what exactly, friends, we will be calling it as a morphogenesis. Next, very important, all functions are important, of course, you have to remember all. Next one, chloroplast maturation. What do you have to understand by this particular chloroplast maturation actually? Friends, please listen to me very carefully. Cytokinin will be helpful in the conversion of the etioplast to the chloroplast. Everyone listen to me. Cytokinin plays a crucial role in the conversion of the etioplast to the chloroplast by stimulating the chlorophyll synthesis in the etiolated seedlings. I have told to all of you many times, what do you mean by the word etioplast? Friends, etioplast means, friends, they are dark grown seedlings which are growing in the dark. So my friends, if this particular etiolated seedling or the etioplast, if it wants to perform the photosynthesis, compulsory it require what? It require the chlorophyll. Whenever there is a synthesis of chlorophyll occur in the etioplast, friends, the pictures are not clear. The etioplast is basically converted into the chloroplast. How? Just by the synthesis of chlorophyll. And friends, how does this particular chlorophyll synthesis get stimulated? Of course, friends, with the help of what, friends? With the help of this particular cytokinin. In a simple word, cytokinin stimulating the chlorophyll synthesis in such a way that it is going to convert the etioplast to the chloroplast. Simple point. Then the last and important function, friends, cytokinin promotes cell expansion in leaves also, cotyledons also. So friends, in order to observe or in order to understand how the cytokinin is enlarging the leaves and cotyledons, we have done the small experiment on the Arabidopsis thaliana. You know, right? What are Arabidopsis thaliana? Jasmine-like seedling. It's not jasmine, friends. We will read in the developmental biology. Arabidopsis thaliana. These are all the seedlings and leaves. All the students listen to me. T0 represents time 0. T3 represents after 3 days. Friends, all of you please kindly look into the diagram. This is the cotyledon and this is the leaf. So friends, when you keep the cotyledon and leaf without cytokinin, you only try to understand there will be no expansion at all. even though you keep for 3 days also, 4 days also, 5 days also, without cytokinin, they are not at all expanding. Later what they have done, friends, to the same seedlings and leaves, they have added the gelatin. What is gelatin? Naturally occurring cytokinin. So when they add the gelatin, what is happening? Obviously, look into this. Gelatin is expanding the cotyledon. Gelatin can also expanding the leaf. How? Of course, by a simple mechanism of cell elongation. Just they allow the cell to get elongated. By elongating the cell, they will allow the leaf to be expanded and cotyledon also to be expanded. So friends, look into the picture carefully. We studied so far all the functions of the cytokinin. I'll just summarize in 1 minute. Cytokinin will show the positive growth in the shoot, negative growth in the root. But due to the mutation, it will show the opposite. That is the positive growth in the root and negative growth in the shoot. And cytokinin also plays a key role in the cell cycle regulation by activating the cyclin D. Then the next very important statements what we read, cytokinin delay the senescence always. Next point, cytokinin, friends, inhibit the apical dominance, whereas auxin is promoting the apical dominance. That's why, my friends, we can call cytokinin and auxin both are antagonistic to each other. And I also said it to you, cytokinin also plays a key role in the morphogenesis, that is the development of a shoot growth majorly. And not only that, cytokinin convert the etioplast to the chloroplast by stimulating the synthesis of a chlorophyll. At the same time, it is also involved in the leaf expansion and cotyledon expansion. That's all. These are all the functions. If you are not getting a question from the function, of course, from where we will get the question, friends? Undoubtedly, we are going to get the questions from the signaling. Signaling is little confused. I will try to make it extremely simple. I'm just requesting all of you, listen my words carefully. Now, every student remember, in the cell signaling we have discussed many times. Oh, sorry, I did not teach cell signaling to you. In the cell signaling you read many times. What kind of enzyme linked receptors that can be used by the hormones? Tell me, Saima, very quickly. Tell me, Reens, Sneha, Sahani. What are the name of the enzyme linked receptors that can be used by the plant hormones or the growth factors present in the plant hormones? Quickly, friends, any one of you? Oya, Priya, Lakshmi, Sadguri, anyone? My question is, what are the name of the enzyme linked receptors that are used by the plant or the growth factors present in the plant or hormones present in the plant? Anyone? Quickly. Very good, very good. No, Sumaya, it's a RTK, there is no doubt about it, but not receptor tyrosine kinases, dear. Plants majorly uses receptor histidine kinases, right? RHKs. See, we are having different type of enzyme linked receptors there, right? We are having receptor tyrosine kinases, we are having receptor serine kinases, we are having receptor threonine kinases, and my friends, even we are having receptor histidine kinases also. Let's take my body. In our body, human body, we are going to read in the cell signaling crystal clearly. Most of our growth factors, they start using what? Receptor tyrosine kinases. In the plant, most of the hormones and growth factors, they start using the receptor histidine kinase receptors in order to show their action. Every one of you listen. More than 3 times in the 2015 also, 2017 also, 2018 also, in part B as well as in the part C, they used to raise the statement about 2-component signaling system. Every student listen my words carefully. If they ask you a question, which of the following hormones, not hormone, hormones, which of the following hormones are involved in the 2-component signaling system, or which of the following hormone uses the 2-component signaling system, I want you people to give the answer ethylene and cytokinin. But they have asked only one hormone. They have given 4 hormones and they asked which of the following hormone majorly uses the 2-component signaling system. They have given both ethylene also and cytokinin also. Then undoubtedly friends, I want you people to choose cytokinin only. But 99.999%, if they given the cytokinin, they won't give the ethylene. If they give the ethylene, they won't give the cytokinin. Don't worry about that. Okay, all of you please try to understand. I will explain in a simple way. Just listen to me for 10 minutes continuously. Focus. There are two different type of the 2-component signaling systems are there. One we call it as a simple 2-component signaling system. Second one friends, we will be calling it as the phosphorylated 2-component signaling system. One is simple 2-component signaling system. Second one is the phosphorylated 2-component signaling system. Let me explain you all in a simple way. This is the sensory histidine kinase domain and it is called as a response regulator. That's why we can call the 2-component. This is one component, this is the second component. All the students listen, look into the diagram very carefully friends. First, who will come and attach us? This is input. To the input, who will come and attach us? Ligand. Here, what is the ligand? Hormone only, right? Cytokinin. Don't confuse. You will understand in the next slide. When the cytokinin associated with the input, we have to show the response of cytokinin, right? Of course, we have to show. For that, the information will pass from input to the transmitter. Friends, how does the information is passing from input to the transmitter? Via phosphorylation. From the transmitter, the information reaches to where? Receiver. And finally, receiver send the information to output. That output ultimately show the response of this particular ligand or a hormone. What was that? Cytokinin. Now, this is what I can call simple 2-component signaling system. Now, every students, please try to look into this friends. Now, we can call the phosphorylated 2-component signaling system. What is this particular phosphorylated 2-component signaling system friends? Every one of you understand. Same. Here sensor histidine kinase is there. Here also response regulator. No doubt. Same thing is there. But along with that, here there is another intermediate is also there. That's why we can call the hybrid sensor histidine kinase. Let me tell you friends, why do I call it as the hybrid sensor histidine kinase? There is a presence of input. Now the ligand will come and attach us. Then input to send the information to the transmitter. Now friends, to the transmitter only receiver associated. But it is not like this, right? In the before, here itself receiver is associated. Now receiver sending the information to the intermediate. Now the intermediate will send the information to the response regulator. Finally, output show the response. Don't confuse with this. Okay, listen my words very carefully once again. This is the hybrid sensor histidine kinase domain. It is having input, transmitter, receiver and intermediate, finally output. Very simple point it is. And friends, actually every student listen to me. This particular hybrid histidine sensor kinase domain was initially identified in the Arabidopsis thaliana. That's why we can call it as AHP. AHP full form is what friends? Arabidopsis histidine phosphotransfer protein. You're going to read that. We can call it Arabidopsis histidine phosphotransfer protein since it is discovered in the Arabidopsis. Very simple. Now, it's extremely important to remember the signaling. I will repeat it twice in a simple way. I'm requesting all of you pay utmost attention. Very important signaling is, there are two things you have to remember. Whenever you are reading the auxin also, I told you the same thing. Whenever you are reading the gibberellic acid also, I told you the same thing. If you want to remember the signaling of a hormone, you have to remember what happens in the presence of hormone. You have to understand what happens in the absence of hormone. That is also you people has to understand. If you understand what happens in the absence and presence, signaling is very easy. Now let's try to understand friends. First, what is happening in the presence of hormone? Suppose friends, when the cytokine is present, obviously the response will be there, undoubtedly. Everyone listen, students, everyone look into this. This is what exactly we will be calling it as a sensor histidine kinase receptor. Everyone, this is called a sensor histidine kinase receptor. having extracellular domain, having transmembrane domain, and friends, cytosolic domain. You people no need to remember that also. What I want you people to remember here, cytokinin will come and associated, right? Suppose this is the cytokinin. Friends, wherever the cytokinin will be associated, that domain only we will be calling it as the chase domain. What do you call it as, friends? We call it as the chase domain. Do not forget that. Friends, the moment cytokinin will come and associated with the chase domain, you know immediately what is going to happen. This is input. This is the input. Then it will send the information to the receiver. How? With the help of phosphate group. Of course, then everyone understand. I told you now, friends, there is an intermediate is present. What is that? Arabidopsis histidine phosphotransport protein. Now the information is reached to the intermediate. That is the AHP. Now that AHP through the nuclear pore, it enter into the nucleus. Simple point. Now finally what domain is there? Receiver domain is there here. This is the one which can show the response. Now this AHP will come and associated with the receiver domain, and it will activate two genes. One we called, friends, type A ARR. Another one we called, friends, type B ARR. What do you mean by the word ARR? Arabidopsis response regulator. What, my friends? Arabidopsis response regulator. All the students, please kindly look into the diagram very carefully. Whenever AHP will come and attaches to the receiver domain, first it will activate what, friends? First it will activate the type B ARR. Now type B ARR will undergo the transcription and translation. Then later they will activate the type A ARR. Now the type B ARR and the type A ARR together, they are going to activate the cytokinin. They can show the response of cytokinin. So I'll quickly summarize for all of you. Everyone listen. Whenever cytokinin will come and attaches to the chase domain of the histidine kinase receptor, the information is passing with the help in the form of phosphate group. So information will come from input to the transmitter to the intermediate. What is intermediate? AHP. How does the information is passing? In the form of a phosphorylation. Now the intermediate AHP enter into the nucleus. It will come and attaches to the response regulator. How? By associating with the receiver domain. Now the receiver domain activate the type B ARR response regulator, type A ARR response regulator. Type B and A together going to bring the response of cytokinin. And every student has to remember for the exam, what is this histidine kinase receptors called as? There are many names are there. We can call CRE1, AHK2, AHK3. CRE1, AHK2, AHK3, we have given the names based on the which plants they are present. But I want you people to remember, normally CRE1, AHK2, AHK3 are the most common type of the histidine kinase receptor used by the cytokinin. Listen to me carefully. CRE1, AHK2, AHK3 receptors most commonly used histidine kinase receptor by the cytokinin. That ends the hormone cytokinin. Now anyone can ask any kind of a doubt from the hormone cytokinin. The question won't come beyond that level, I am very, very sure. Is it clear? Everyone say quickly, my friends. Is that clear? Is that clear? Quickly, friends. You people ask me any kind of a doubt. Okay, I hope it's clear for every one of you. Okay, so friends, what are the various hormones that we have completed, friends? We have completed the hormone auxin, we have completed the hormone gibberellic acid, and we have completed the hormone that is the cytokinin. Now friends, what are the two other hormones that we are left with, my friends? We are left with the hormone called as abscisic acid also, as well as, friends, we are left with the hormone ethylene. Both are important. My friends, actually hormones, all 5 hormones are extremely important. There is no doubt about it. Once again, as I told you, shortcut. If you want to remember the hormones, what is the shortcut? Comparing one hormone with another hormone. When you compare, it will become extremely easy. Now listen, when we started reading the auxin, what I told to all of you, remember as a one word, auxin is the rooting hormone, I told to all of you. Whenever we are reading the cytokinin, what did I say to all of you to remember? Cytokinin is the shooting hormone, I told you. We also read the gibberellic acid. What is the one word I told you to remember? Gibberellic acid is major hormone involved in the seed germination. Same way, when you heard the name of a abscisic acid, 2 points has to strike in your mind. What are the 2 points has to strike in your mind? First point, abscisic acid is the stress hormone, which means which is going to release it in the plant body during the stress condition. You know, right? When we are under the stress condition, in our body adrenaline will release or cortisol will release majorly. Same way, whenever plant is facing lot of stress condition, to tolerate all the stress condition, plant starts releasing the abscisic acid. That is the first unique statement. Second unique statement you have to remember, this is the powerful growth inhibitor. It's not a growth promoter. See, I always remember in this way. By that I can remember for longer time. Now quickly I am summarizing for all of you. Auxin, what is the name of the precursor that is involved in the biosynthesis of auxin? We read tryptophan. Very simple. I will remember like this only, friends. And cytokinin, what is the precursor that we have studied for the cytokinin? We studied DMAPP. Just now we've completed. And we also studied the precursor for the gibberellic acid. We studied very clearly. That is the GGPP. Now you only try to understand what is the precursor for the abscisic acid. Every student has to remember. That is the IPP. We read many times, right? Isopentenyl pyrophosphate, which is how many carbons, friends? Isopentenyl pyrophosphate is the 5 carbon containing compound. Please kindly don't forget that. This is the one which is acting as the major precursor. Please kindly don't forget that. Now, one more interesting point. What is the name of the naturally occurring auxin that we have studied, my friends? We discussed very clearly naturally occurring auxin. I said IAA, indole acetic acid. And what is the naturally occurring cytokinin? I told to all of you, gelatin. Please remember, in this way concept will be very, very clear. And friends, what is the name of the major gibberellic acid most active form? We already discussed it, right? GA1 is the most active form of the gibberellic acid. Now friends, please listen to me. The most naturally occurring abscisic acid in the plant body is the zeaxanthin. See how easily we can remember. We can call it as what, friends? Zeaxanthin. Zeaxanthin is the naturally occurring abscisic acid. Gelatin is the naturally occurring cytokinin. Please kindly don't get confused. That is gelatin, this is zeaxanthin. All are important for the exam. Now let me tell you, how does this particular abscisic acid got discovered? Every student listen, friends. Abscisic acid is a sesquiterpene. Sesquiterpene means what? 15 carbon containing compound. It is derived from IPP, which is a 5 carbon. So this 5 carbon containing compound only giving rise to this 15 carbon containing compound that we are going to read in the very important chapter, that is the secondary metabolites. But I want you people to remember, I will ask you undoubtedly, I will ask you while we are reading the secondary metabolites. Listen my words and remember this. Sesquiterpene is a 15 carbon containing compound. Friends, you know how the abscisic acid discovery occurred? It was really interesting discovery. Everyone please listen to me. In the year 1960, 3 individual scientists, listen to me, in the year 1960, 3 individual scientists from 3 different laboratories, 3 individual scientists from 3 different laboratories, from 3 different places, they have identified 3 type of inhibitors. Different people, different places, different laboratories, and they identified the 3 different type of inhibitors. One we called Darmin, another one we called abscissin 2, another one we called, my friends, inhibin. Every student, please listen to me carefully. After few years, it was surprisingly found that, listen to me, after few years, it was surprisingly found that these three inhibitors are structurally and functionally absolutely same. Three are absolutely same, both structurally and functionally. So based on these three names, they have given a name abscisic acid. Don't forget the point. Now here comes another unique statement you have to remember. When we read the auxin, friends, you only quickly recall, when we read the auxin, we read that auxin is synthesized in the root. Cytokinin, I'm so sorry friends. Auxin is synthesized in the shoot, travels to the root. Cytokinin synthesized in the root, travel to the shoot. And what did I tell you about the gibberellic acid? It is majorly synthesized in the seed. Okay, now every one of you try to understand the another most important point, friends. I think CSIR NET result out. I think, okay, you can check it. Okay, after the class. So friends, whenever you are reading the abscisic acid, I want you to remember abscisic acid is the ubiquitous plant hormone. Abscisic acid is acting as what? Ubiquitous. Friends, what do you mean by the word ubiquitous plant hormone? It can be synthesized everywhere in the plant body. Everywhere in the plant body. Which means, let's take an example, bacteria. What is the speciality of bacteria? Bacteria grow everywhere, right? Bacteria grow at high temperature, high pH, low pH, high pressure, low pressure. Everywhere they can grow. Same way, friends, abscisic acid also in the plant body, they grow everywhere continuously. Please kindly do not forget the statement. Abscisic acid is the ubiquitous plant hormone synthesized in the plant body everywhere. But you should not think when I use a word abscisic acid is synthesized everywhere in the plant body, you should not think that abscisic acid is releasing in the highest concentration. If the abscisic acid released in the high concentration, plant will die immediately. No, abscisic acid released in the all parts of the plant body at a very low concentration. Kindly don't forget that, friends. And next one, where does this particular abscisic acid biosynthesized? Every parts of the body. And what I said to you, it is a sesquiterpene, friends. And what did I say to all of you? Naturally occurring abscisic acid is what? Zeaxanthin. Every student has to remember. You don't need to remember the structure. This was asked part B question. What is the part B question they have asked is, they asked a question, which of the following form naturally occurring abscisic acid can be present? Always remember, naturally occurring abscisic acid zeaxanthin, it is always exhibited in the cis form. There will be a cis zeaxanthin is there, trans zeaxanthin also there. Or are cis type of abscisic acid or trans type of abscisic acid. So mostly, friends, the zeaxanthin will be cis time, which is extremely biologically active. So quickly try to understand, friends. Abscisic acid can be transported through both xylem and phloem through the entire plant body. Naturally occurring abscisic acid is the zeaxanthin. Precursor used is the IPP. Now, friends, let's start understanding most important things, that is functions of the abscisic acid. First function, I have clearly told to all of you, abscisic acid is the hormone which is a stress hormone. Now every one of you listen. Suppose example, the transpiration rate is very high. You only tell me, friends, when the transpiration rate is high, what happens actually? Stomata get open and entire water loss will occur continuously. If the water loss is occurring continuously, there is a great chance that plant may die immediately. So we have to overcome that. How can we overcome that? By closing the stomata in order to prevent the water loss by the process of transpiration. So every student listen, it induces the stomatal closure to prevent the water loss by the transpiration. In the next slide, I will explain to you how exactly abscisic acid is involved in the opening and closure of the stomata. Don't worry about that, but just listen. Abscisic acid plays a key role in the stomatal closure. Next point, Abscisic acid is the another important hormone promote the seed dormancy. Every student has to listen this important statement. Abscisic acid promoting the seed dormancy. When abscisic acid promote the seed dormancy, there will be a no fertilization. Fertilization never occurred. Whenever there is a no fertilization, friends, there is a no seed germination. Simple point. It promote the seed dormancy means no fertilization. Dormancy, friends, how can you define the word dormancy? Dormancy or dormant. Dormant is the period where there is a no fertilization. When there is a no fertilization, then there is a no seed germination. Plant will reach the desiccation condition also. What is desiccation? Desiccation is the condition where there is absolutely no water content in the plant body. Plant is going to reach zero water condition. So that kind of conditions can be promoted by the abscisic acid. My friends, all the students listen to me. Abscisic acid promoting the seed dormancy, whereas my friends, gibberellic acid is going to inhibit the seed dormancy. Opposite. Once again, every student listen to me. When abscisic acid promote the seed dormancy, no fertilization. Since gibberellic acid is inhibiting the seed germination, there is a fertilization and then there is a seed germination. Just opposite functions of the abscisic acid shown by the gibberellic acid. Once again, we can say abscisic acid and gibberellic acid also act antagonistic to each other with respect to the seed dormancy. Please kindly do not forget this particular statement at all. Both are acting oppositely. That's the reason you have to understand all the hormones present in the plant body will do the interplay. What do you mean by the word interplay here? One hormone action can be enhanced by the another hormone, or one hormone action can be inhibited by the another hormone. Never ever forget that particular point. Now the third and unique statement, and don't confuse with the third statement actually. It promote the leaf senescence independently of ethylene. Never ever forget this. It promote leaf senescence independently of ethylene. Friends, in the starting of today's class also I told you na, what is the major hormone involved in promoting the senescence? Ethylene. We will read next. What is the major hormone that delay the senescence? We already studied cytokinin. So ethylene enhance the senescence, cytokinins delay the senescence. Then why we are reading abscisic acid? What I want to convey here, abscisic acid is also promote the leaf senescence, not in all the plants. Certain plants, especially citrus plant, but not all the time. Whenever ethylene levels are extremely low, whenever ethylene levels are extremely low in the plant body, only during that time, friends, abscisic acid is going to promote this particular leaf senescence. Don't forget this. I hope it's clear for every one of you till the third point. So friends, remaining thing we will continue in the tomorrow class. Okay. So friends, tomorrow I am saying to you 99.99% I will take the class at 5:30 AM only. Actually, yesterday and today there was a no power. Okay, that is the reason I couldn't take. 99.99% 5:30 AM only. Don't worry at all. Sharp 4 in between 4:30 to 5 AM I will give the message in the telegram that the class is at 5:30 AM. If the class is at 8, mostly I don't take in the evening times. I will take at the 5:30 AM only, because due to the power problem only I have taken this. Okay, I will take 5:30 AM only, but I will inform you by 4:30 itself. Please kindly don't neglect. And one more important point I am highlighting here. By 31st of March, these chapters will be completed. Unit 3 will be completed. Unit 6 will be completed. Right now I am giving, once again I am giving information. Either 2nd April or 3rd April, you all people has to write a test on this particular unit 3 and unit 6 also. Please kindly prepare for that. Later, I already told you, in the month of April, I will finish 3 chapters at a time. You have to attend many number of classes. So be prepared for that. Okay friends, that's all for now. I hope the discussion is little useful to all of you. Please prepare well. I'll see you in the tomorrow morning class. Bye bye.